You can see how it is copied into mRNA and carried out to the cytoplasm, where rRNA and tRNA use it to build proteins. Though all the cells in an organism contain exactly the same DNA, different types of cells in a multicellular organism take on different forms and functions. What causes a blood cell to be different from, say, a nerve cell is that various factors cause them to transcribe different sections of the DNA in the nucleus into mRNA, thus causing the cells to produce different proteins and look and function differently. Any given cell in an organism thus only transcribes those genes relevant to its function in the organism. For example, stem cells in bone marrow that produce red blood cells constantly transcribe the genes necessary for the production of hemoglobin, which is critical in a red blood cell's role of carrying oxygen to and carbon dioxide away from other cells in the organism. On the other hand, muscle cells probably never transcribe those genes, but frequently transcribe the genes for actin and myosin proteins crucial to the contraction of muscles. Thus, in any given cell, transcription typically only copies the DNA of selected genes into RNA. During transcription, only one strand of DNA is transcribed into RNA. Although both sides of the DNA and a chromosome contain genes, the code for any given gene is usually totally on one strand or the other. The strand being transcribed into RNA is often referred to as the sense strand, as it is the one of the two strands that makes sense. The process of transcription is divided into three parts: initiation, elongation, and termination. Transcription, or as it is often referred to, RNA synthesis, begins with initiation. A promoter region lies just upstream from the beginning of each specific gene. RNA polymerase, the enzyme that carries out RNA transcription, recognizes the unique nucleotide sequence of the promoter region of the gene to be transcribed and attaches itself to the DNA there. After it attaches itself, the RNA polymerase starts forcing the strands of the DNA to open up and separate at the beginning of the gene. This starts the process of elongation. The RNA polymerase moves along the sense strand in the direction dictated by the orientation of the sugar phosphate molecules that form the backbone of the strand, just as DNA polymerase did in DNA replication. Using free RNA nucleotides in the nucleus, it builds a single complementary strand of RNA using the same base pairing rules as used in DNA replication, except that uracil, rather than thymine, is now paired with adenine. Unlike DNA replication, however, the base pairings are not permanent, but rather after 10 or so nucleotides have been added to the growing RNA strand, the beginning molecules begin to separate and form a tail that extends out from the DNA. The final step in transcription is termination. The RNA polymerase stops elongating the strands of RNA and releases it. The RNA polymerase then disappears, and the strands of DNA rejoin to one another. The newly formed RNA will leave the nucleus through pores in the nuclear envelope. If it is mRNA, it will attach to a ribosome, where its genetic code will be read and a protein synthesized. If it is rRNA, it will join with other rRNA strands and a variety of proteins to form a ribosome. If it is tRNA, it will bind to amino acids and carry them to a ribosome in order to create the string of amino acids that will form a protein. The process of reading the genetic code on an mRNA molecule and converting or translating it into a protein is fittingly enough called translation. Translation occurs in ribosomes that, as mentioned earlier, are made up of rRNA and proteins. Ribosomes consist of two parts, a small subunit and a large subunit. The small subunit is made up of one rRNA and about 30 proteins. The large subunit consists of 3 rRNA and 45 to 50 proteins. The large subunit also contains an enzymatic region that catalyzes the addition of amino acids to growing protein chains, and two sites, usually referred to as P and A, that temporarily attach to tRNA during translation. Translation, like transcription, occurs in three stages: initiation, protein elongation, and termination. Initiation begins when two codons of mRNA and several protein initiation factors bind to a small ribosome subunit. The first codon is always the AUG, or start codon. In this example, the second codon is AGU. A tRNA with the appropriate amino acid bound to it will attach to each of these codons. There are many different kinds of tRNA, at least one for every amino acid, but all have a shape somewhat like a three-leaf clover. Amino acids are bound to the stem of tRNA by enzymes that recognize each specific tRNA and attach the correct amino acid to their stem. The top of the central leaf of each tRNA has three exposed bases that form a structure called an anticodon. The anticodon of each tRNA is complementary to the codon of the mRNA that specifies the amino acid attached to that tRNA. AUG specifies the amino acid methionine, and its complementary or anticodon is UAC. So a tRNA carrying methionine and the anticodon UAC form hydrogen bonds with the start or AUG codon. After that occurs, the large subunit attaches to the small subunit, and simultaneously the tRNA attaches to the P site on the large subunit. Now that the ribosome is completely assembled, it is ready to begin the process of protein elongation. A tRNA with the anticodon UCA, the complementary codon for AGU, now moves into site A, which is adjacent to site P on the large subunit. The catalytic region below and between sites A and P on the large subunit break the bond between the starter amino acid, methionine, and its tRNA, and uses the released energy to join the methionine to the serine carried by the second tRNA. The now empty start tRNA drops off of its site P, and the ribosome shifts one codon to the right on the mRNA, shifting the second tRNA with two amino acids now attached to it into site P. A new tRNA with an appropriate anticodon and amino acid now moves into site A. The catalytic region breaks the bond holding the two amino acids to their tRNA and joins them to the new amino acid, in this case glycine, in site A. The empty tRNA in site P drops off, and the ribosome again shifts one codon to the right. The process repeats like this until a long protein chain is formed. Termination occurs when near the end of the mRNA, a stop codon, UAA, UAG, or UGA, is reached. No tRNA bonds to the stop codon. Rather, termination factors cut the completed protein from the last tRNA, releasing it from the ribosome. The long protein chains, built through the process of elongation, then fold into their characteristic shape. Some of these proteins immediately go to work in the cell, carrying out structural roles or catalyzing chemical reactions. Other proteins, such as various digestive enzymes, remain inactive until they are released outside the cell, where factors in the external environment turn on their active sites and make them fully operational. The proteins created as a result of the processes of transcription and translation, in large part, define the characteristics of every living organism, including we humans. The conversion of this information stored on strands of DNA into the structural and catalytic proteins that form the essence of all living organisms populating the Earth is the most fundamental and important process in the living world. By understanding and being able to manipulate the process by which information stored on long molecules of DNA is converted into living, breathing organisms, we are entering an era in which we will see unprecedented advances in biotechnology, medicine, agriculture, and other areas of human endeavor. However, this new understanding will also ask us to make important ethical and moral judgments about how far we as humankind should go in altering and/or perhaps even creating life. The future of all life on Earth will depend on how human society answers these important questions about turning molecules into life.